Welcome back, everyone. Our last video was on transcription, and we ended on nuclear export through the nuclear envelope, the double membrane that separates the nucleus from the rest of the cell. I also want to correct something I said about the last video. Transcription, or initiation, elongation, and termination. Although I believe you can use that to describe transcription, I think it's better off if you use it to describe translation. Just because it's more official, I think. So, what happens after the mRNA molecule with the 5' and the polyadenosine tails leaves the nucleus? So, after the newly transcribed mRNA is exported from the nucleus, it needs to bind to a ribosome to initiate translation. So, what is a ribosome? A ribosome is an RNA, is composed of RNA and a number of miscellaneous proteins that help it function. Ribosomes read codons. So, as we know, the mRNA is composed of a long chain of nucleotides. Every 3 nucleotides is considered a codon. I've attached a image here of, and I'm going to go through this so that it's a little bit more clear. So if you look at the chart here, it says the first letter is going to be here. So we have U, C, A, and G. The second letter is going to be here. So we have U, C, A, and G. There's a total of 64 codons, but only 61 of them actually code for amino acids. Amino acids, we have phenylalanine here. So it shows you the first letter, the U, the uracil, and the uracil make phenylalanine. The third amino acid or the third nucleotide does not matter as much. It's called the wobble effect, and it can signal anything. But the reason it's very non-specific is because it limits the amount of mutations that can happen, because it gives you a little bit of wiggle room, wobble effect. So just remember, the third nucleotide is not as significant as the first two. The first two are what determine the amino acid. And we have phenylalanine, leucine, isoleucine, methionine, valine, alanine, threonine, proline, serine, tyrosine, histidine, glutamine, asparagine, glycine, aspartate, glutamate, glycine, arginine, serine, arginine, tryptophan, and cysteine. So if you want to learn a little bit about amino acids, which are the building blocks of proteins, hence translation, we translate mRNA into protein. Proteins are composed of amino acids, and that's what these nucleotides are codons. These groups of 3 nucleotides can code for. So something to note, AUG is the start codon. Me growing up, I played a lot of Call of Duty, so I always thought that the AUG was a good starter gun. So that's a really weird connotation, or association, but that's what I associated AUG with. The stop codons I just kind of memorized: UGA, UAA, and UAG. They stand out, so if you ever see them in a testing center, just make sure you're like UGA, UA, or UAG. It's basically like, it's AUG, but like different. There's plenty of mnemonics on YouTube that you can use to memorize those 3, but everyone's going to memorize things differently. So amino acids form proteins through something called a peptide bond. And a peptide bond is between a carboxyl group, a carbon and an oxygen, double bonded to an oxygen, and an amine, which is a nitrogen compound. There's primary amines, secondary amines, tertiary amines. In terms of proteins, most of them, except proline, are going to be primary amines used to form peptide bonds with the carboxyl group. Just because, and primary means like there's only one carbon. So in this example, this would be a carbon, this would be a hydrogen, this would be a hydrogen. So it's primary because there's only one carbon bond. And here the electrons we drew earlier, they're performing a nucleophilic attack. So if you've learned about dipoles, we know that oxygen is more electronegative than carbon, meaning it's going to pull the electrons with a greater force. It almost has it's closer to a full octet. I don't have a periodic table right now, but oxygen is more up and to the right than carbon on the periodic table. If you don't know, electronegativity tends to trend upward and to the right. So fluorine would theoretically be the most electronegative, theoretically. And that creates a dipole. The nitrogen is an electron donating group, so it's going to donate that negative charge to the partial positive charge on the carbon due to the dipole with oxygen. You can research that more if you want. It's a pretty important topic for chemistry, but since we're just going over translation, because I want to turn this channel into an immunology and cancer oriented channel where I read like scientific articles and talk about them and think about where we're going. But we have to get through these basics first, and it's good to it's good to refresh our memories. So inside the ribosome, there's something called tRNA. tRNA stands for transfer RNA. Starting here, we'll have like the 5' end of the tRNA. Remember this, don't get this confused with mRNA. mRNA is messenger RNA, which takes the transcribed, basically the mRNA that we transcribed in the nucleus using the template strand and put a 5' and a polyadenosine tail on it and shipped it out of the nucleus. The tRNA is located inside the ribosome. And this is what's going to help us translate. So tRNA has 5 components if you include this little horn right here. I'll just circle it and then undo. So this is going to be the amino acid binding region. Amino acyl tRNA synthetase. It utilizes GTP, guanosine triphosphate. It conducts a reaction where it breaks, makes guanosine diphosphate and inorganic phosphate, which is a lone phosphate group, because it requires energy to bind things. Whenever you create a bond, it requires energy. And whenever bonds break, they release energy. But that's chemistry. I'm sure we'll get there at some point. But back to this. So tRNA, there's an amino acid binding site. It takes energy, uses the enzyme amino acyl tRNA synthetase. We have the T arm. This connects the amino acid right here. This can be a generic amino acid. So amino acid, we just use AA for amino acid. And connects it with the anticodon. The anticodon down here is going to be, so this region right here is the anticodon region. It's going to be the thing that matches up with the mRNA codons. So this little horn I mentioned earlier is like the variable region, I believe it was called. It's different for each ribosome and doesn't really hold any significance for the sake of this lesson. Then the D arm on the right here verifies, it basically verifies the identity. I don't know why that doesn't say identity of the codon and the anticodon, and it makes sure they match. If it matches, it says good to go. Let's start translating. So that's basically the tRNA structure. And these, I didn't draw them, but there's basically a bunch of nucleotides. Here. This is not to scale, but there's generally quite a few of them. For the sake of simplicity, we're just gonna make sure they're there, but it's not too accurate of a depiction. Oops. All right, moving forward. Okay. So, what kind of ribosomes are there? So, as we know, they're prokaryotic ribosomes before the nucleus cells, and they're ribosomes, and then eukaryotic ribosomes that are in eukaryotic cells that have nuclei. Sorry. So starting with the prokaryotic ribosomes, they have a 30S and 50S subunit, which come together and make a 70S ribosome. S stands for Svedberg, and did some digging, and apparently it's a unit of density. If enough people leave comments and ask, I can research more about what it actually, like why 30 and 50 make 70 and 40 and 60 make 80. I don't know, but it's probably important. Another day. But yes, the 30 and 50 subunits of the prokaryotic ribosomes make a 70S ribosome, and eukaryotic ribosomes are composed of a 40S small subunit and a 60S large subunit, which come together to make an 80S ribosome. This is important because prokaryotes like bacteria and archaea utilize different ribosomal subunits, and this can be used as a clinical target for medications such as aminoglycosides and tetracyclines, which attack the 30S subunit, or another brand of antibiotic called the macrolide, which target the 50S subunit, the large subunit. Aminoglycosides, they lock the starter codon AUG methionine in the initiation site and inhibit further translation. That doesn't make sense now, but it will. And they incorporate the incorrect amino acid and they lead to premature termination. So basically, they stop translation at its tracks at the beginning because they inhibit the ability of AUG to move from the initiation site. Second, they can interfere with gram-positive lipopolysaccharide layers. So in these prokaryotes, in gram-positive, and gram-positive is just from the gram stain. There's a certain stain called the gram stain that you can use, and if the bacteria has a cell wall, it stains. Like, I think it's purple. It's either blue or purple. I'm not sure at the moment. I can look into that too. But if it's gram-positive, aminoglycosides interfere with that lipopolysaccharide layer and increase the permeability. It's usually used in a antibacterial cocktail because it increases permeability to other antibiotics in gram-positive bacteria. Types of aminoglycosides are streptomycin, neomycin, and gentamicin. Next, we have tetracyclines. Hinders the 30S, the small subunit of prokaryotic ribosome, by inhibiting amino acyl tRNA synthetase. As mentioned earlier, amino acyl tRNA synthetase is what initially connects the amino acid with the tRNA via guanosine triphosphate. When amino acyl tRNA synthetase is catalyzing the reaction, it breaks up guanosine triphosphate into guanosine diphosphate and an inorganic phosphate. These breaking of the bonds release energy, which allows the amino acid to bind to the tRNA, because, like we said, anything that bonds requires energy and generates that energy from the breaking of the phosphate bond. This induces a bacteriostatic state because the amino acid cannot be charged unless elongation is halted. So, before we move on, I think it's important to say that in these ribosomes, in the large subunit, so in prokaryotes, this would be the 50S subunit, and in eukaryotic ribosomes, this would be the 60S subunit. They all have an A site, a P site, and an E site. The A site stands for amino acyl site. It's where the mRNA initially arrives, and it's red. The peptidyl site, where the correct anticodon and amino acid is added. Well, that's not true. It's just where the anticodon is lined up. It's added in the A site, amino acyl site, but we'll get to that later. Right now, I just need to know that A is equal to amino acyl site, P is equal to peptidyl site, and E is equal to the exit site, where things get shipped off, or like thrown back into the abyss of the cytoplasm. Um, so, initiation is the first step. We are going to get through this. Shine-Delgarno sequence, purine sequence. So if you remember in the very first video of DNA, purines, basically the rings, A and G, they are silver, and you'd rather have a lot of silver rings. So adenine and guanine. It's a large amount of adenine and guanine, a sequence about 8 nucleotides upstream of the start codon. So the start codon is AUG, which codes for methionine. In prokaryotes, it encodes for N-formal like the letter N, formal methionine. And the Shine-Delgarno sequence, it basically signals for initiation factors in the 30S subunit, the small subunit of prokaryotes, to recognize the sequence and initiate binding of N-formal methionine. As required with all binding, GTP is used to find, well, not all binding. In this case, it's GTP. Another molecule used to bind things is ATP, but in this case, it's GTP. And ATP is just adenosine triphosphate. GTP is guanine triphosphate. Is used to, so GTP is used to bind N-formal methionine to AUG. Because it takes energy to form bonds. When bonds are broken, energy release is a little redundant. But hey, repetition never hurt anybody. So, as mentioned earlier, amino acids bind to tRNA, which is located within the ribosome, specifically in the P site, through amino acyl tRNA synthetase and the conversion of guanosine. Diphosphate. There we go. Into guanosine diphosphate. The breaking of the phosphate bond creates the energy needed to bind the amino acid, the fundamental building block of proteins, to the tRNA. In eukaryotes, instead of using the Shine-Dalgarno sequence to signal binding of that initial tRNA segment, eukaryotes use initiation factors 4 to line it up, and they use eukaryotic initiation factor 2, which requires GTP to bind methionine to AUG. So basically, elongation and initiation factor 4 starts the process. Eukaryotic initiation factor 4 starts the process. Eukaryotic initiation factor 2 binds methionine methionine to AG. It's not methionine. It's the anticodon. Because we already established that amino acyl tRNA synthetase binds the methionine to the tRNA. So eukaryotic initiation factor 2 binds UAC to AUG. If you need to review the base pairs, I recommend you go to our first video on DNA, or even transcription. Both videos will probably be useful, but I can just add that here so it makes a little bit more sense. UAC, there it is. Boom. So there you have it. You have the anticodon, which is bound to the mRNA strand, which is the strand sandwiched in between the two subunits. And you have the tRNA holding together the anticodon with the amino acid, which is going to make the protein. Okay, so after initiation, you get elongation. So this is the part that's honestly the most interesting, but the most convoluted. And if you are in chemistry, this will be very helpful. But if you're not in chemistry, we can just use non-chemistry terms to kind of explain this. So I'm gonna read the summary and then I'm gonna explain it. So after initiation of the starter codon with the anticodon, because that's what initiation is, it's basically lining up the process. A translation factor, a signaling molecule called elongation factor type 1, bound to GTP, facilitates the binding of the second complementary codon in the A site. So, as I mentioned before, you see this E. Oops. Okay, we're just going to use the pen. We have E. We have P. And we have A. We know that E is the exit site. We know P is the peptidyl site. And we know A is the amino acyl site. And we know that unintuitively, I guess, it goes from right to left. The mRNA strand. So this is the mRNA strand. It's got these codons, which are basically groups 3 of nucleotides, and it gets sandwiched through, and it goes through this little process here, from right to left. So I'm just gonna draw that. Put the 5 over here, so that we know. So the 5 cap is for eukaryotes, and somewhere way over here is a 3 end. 3 prime. Cool. So now we have our directions going from, it's translating from 5 to 3. and is moving towards the 5. Or the 5, it's moving to the left. Yeah. So moving forward, elongation factor 1 is going to signal. So after this initiation step has been squared away, and the codon is matched with the anticodon, and methionine has been the starter codon has been established, you move to the A site where the next codon is going to pair and bring with it the amino acid that has been connected to the tRNA, which actually, hold on, by that reasoning, tRNA would be in the A site and not just the P site. Let me think about this for a second. Okay, welcome back. So I looked it up, and basically I was confused on whether tRNA only worked in the P site. Turns out it works in all three, or it can. So it's not really an issue here. So elongation factor 1 binds to GTP. GTP breaks into GDP and an organic phosphate, breaks the bond, matches the anticodon. In this case, we're using uracil, which codes for lysine in the A site, and it's demarcated by, or it's it's marked here with the AAA. So then, here's where the chemistry comes in. The N terminal, or the N terminus, the amine, is going to attack with the electrons, as noted here, at the dipole. Remember, oxygen's more electronegative and it draws electron density towards it, allowing the electrons of the amine to attack that carboxyl group. So after that, an enzyme called peptidyl transferase. Brings the new peptide bond to the A site, and then you have the beginning of a peptide or polypeptide. So the polypeptide's made in the A site. Then translation factor, known as elongation factor 2, is activated. Elongation factor 2 is often activated by phosphorylation, or GTP again. Not phosphorylation, excuse me. The breaking of the GTP to GTDP and inorganic phosphate, and that moves the polypeptide back to the P, so that the next codon in this mRNA sequence can be bound to an anticodon on the A site. In this, now we're going to go to termination because this video is going long and I'm running out of time, so I'll have to make this another video. But elongation, so after elongation factor 2 signals for the shift of the newly synthesized peptide to shift over to the P site from the A site, the anticodon in the E site is ejected because it is no longer necessary. So the starter codon that we started with, AUG, noted here, gets ejected because it's no longer needed. And now those stop codons, remember there's 64 codons and 61 of them encode for amino acids. The 3 that don't are UGA, UAA, and UHE. These code for release factor, and that binds this codon where the anticodon should be. And immediately it stops translation, signals for cleavage, and releases the peptide from the P site. So this peptide, this polypeptide, noted by the peptide bond between the two amino acids, is ejected through the P site. And stop codons do not have amino acids that they code for. They signal the release factor to bind the stop codon, and this stops translation, signals for polypeptide cleavage, and release of the peptide from the P site. I don't have enough time to get through ribosomal translocation and post-translational modification, so we'll get to that another time. But that is the bulk of translation. Let me see how much time I got. Oh, I think I made 2 separate videos. That's all right. We'll go through that another day. So just going over a review. tRNA, it can go through A, P, or E site, but it's basically the transfer RNA in the ribosome. Big things you're going to want to know here is that the eukaryotic and the prokaryotic ribosomes are different. Eukaryotic ribosomes are larger. And yeah, you're going to want to know that anytime it binds, it uses GTP. Starter codon is AUG. End codon or stop codons are UGA, UGA, UAA, and UAG. UGA, UAA, and UAG. Yeah. Sorry about the disorganization of this video. It was rather late, long. Don't want to make excuses, but I do want to get through these beginning biological concepts. After we do ribosomal translocation and post-translational modifications, I want to do mitosis and meiosis, and then we can get into cancer, which is my strength. So thank you if you enjoyed this video. Thanks for tuning in. Hopefully it wasn't too confusing. I know this wasn't my best organization wise. And yeah, see you on the next one.